Imagine you're a detective solving a medical mystery. Your clues, a patient's symptoms, timing of events, and laboratory results. Your mission? Determining if a medication is truly responsible for an adverse event. This is causality assessment, and today we'll master one of its most powerful tools, the WHO-UMC system. The World Health Organization's Uppsala Monitoring Center created this system to bring clarity to the complex world of adverse event analysis. Born from decades of pharmacovigilance experience, it has become the cornerstone of standardized causality assessment worldwide. Think of it as your compass in navigating the challenging waters of adverse event evaluation. Let's dive into our first and most definitive category, certain. Picture this. A patient starts carbamazepine therapy for epilepsy. 3 weeks later, they develop Stevens-Johnson syndrome, a severe skin reaction. The timing is perfect, typically occurring within the first 8 weeks. Laboratory tests confirm the immunological reaction. When the drug is stopped, the condition improves. Unfortunately, when accidentally re-administered, the reaction recurs, a classic re-challenge scenario. This is what we call a slam dunk in causality assessment. We have 4 key elements: a plausible time relationship, a recognized reaction pattern, positive de-challenge, improvement when stopped, positive re-challenge, recurrence when restarted. Moving to probable likely, let's examine ACE inhibitor-induced angioedema. Consider a patient who develops facial swelling 2 days after starting lisinopril. There's no history of allergies, no other new medications, and the swelling resolves after discontinuing the drug. Why isn't this certain? Because we're missing re-challenge data, and for good reason. We wouldn't deliberately re-expose the patient to risk. However, we have reasonable time relationship, known reaction pattern, positive de-challenge, and no other clear explanations. This is the category you'll encounter most often in real-world practice. Now, let's tackle possible with a case of drug-induced liver injury. A patient on multiple medications shows elevated liver enzymes. Could it be the new antibiotic, the chronic heart medication, or perhaps their underlying condition? This category acknowledges when the timing makes sense, the reaction pattern is reasonable, but other explanations could exist. Think of it as having several suspects in your detective story, all with plausible motives. Finally, let's explore unlikely cases. Imagine a patient reporting memory loss starting 6 months before beginning a new medication. The timeline here tells us this can't be our culprit. Or consider a patient with clear symptoms of viral gastroenteritis who happens to be on a new antihypertensive. Key markers of unlikely cases include implausible time relationships, more probable alternative explanations, and disease patterns that don't match known drug effects. Remember, causality assessment isn't just an academic exercise. Each category decision impacts patient care decisions, safety signal detection, regulatory reporting requirements, and future treatment options.